Okay, good afternoon, everybody, and welcome to this afternoon's webinar on analysing linkage disequilibrium with Ensembl. My name is Emily Perry, and I'm the Ensembl Outreach Project Leader. That means that I run a small team who deal with many different ways of teaching people and telling people about Ensembl. So we deliver various kinds of training, both online, like we're having today, and in person. We also do things like online help and documentation with the people. If you email Ensembl and ask for help, we get back to you. So you'll see if you look at your interface for the webinar, you'll see that we have muted all your microphones. There's a chat box, or it might be called the questions box if you've got a more recent version. If you have any questions throughout the webinar, please type them in there. I have my colleague Erin in the room with me, and she's ready to answer any questions. And if there are any more at the end, we'll answer those as well. There is no threading in it, so please look out for the answer to your question, which should have @ and then your name. That you have to actually type that in. So what we're going to learn today is what Ensembl is, the types of data you can explore in Ensembl, specifically looking at linkage disequilibrium. We have some pages for looking at linkage disequilibrium and also a calculator tool. And finally, we'll talk about my favorite thing, help and documentation. So Ensembl is a genome browser, and genome browsers are very important because they allow us to deal with the vast amounts of data that a genome is made up of. Human genomes are about 3 gigabases. If you look at a wheat genome, I think it's at least 10 times as big as that. So we are talking quite vast amounts of data. And that data looks like this. It is utterly meaningless by itself. It only has any value when you incorporate it together, when you annotate things onto this. So genome browsers are one of the ways that we unlock the code, as it were, of genomic data. So the major ones out there, and there are sort of smaller scale ones as well, are Ensembl, UCSC and NTBI have a couple of genome browsers as well. Ensembl have loads of data. We have genomes with what we call gene builds, which is basically all of the genes plotted onto the genome for over 100 species. We have variation data, and we're going to look at a bit of that when we look at the linkage disequilibrium today. We also have comparative genomics data, including alignments, gene trees, homologs, and we have what we call a regulatory build, which incorporates data from sources such as ENCODE. This is alongside things like gene expression data, functional data. We have different ways to get hold of that data as well. So we have a tool called Biomark for data export. We have tools for data processing. Some of you might have heard of our variant effect predictor, and we'll be looking at another of our tools today, which is the linkage disequilibrium tool. We allow you to display your own data. We have APIs, both a Perl API and a REST API for programmatic access, and we are completely open source. We have an FTP site, our data and code is all on GitHub as well, and it's all free to download and to use as you wish. We have variation data from a number of different sources. This is just listing all the sources where we have variation data for human. So giving you some examples, I'm not going to go through everything on this page, but we have our major sources, dbSNP, who provide accessioning for submitted variants, short variants, from a number of different sources. We get things like XAC and not XAC anymore because that's incorporated into Nomad. Nomad, that's what I meant to say, and 1,000 genomes we get through dbSNP because they accession the data. We also have somatic variations from cancer, from Cosmic. We get phenotypes and things from the GWAS catalogue. We work with Uniprot, so a number of different sources. As I said, I won't go through all of these, but if you go to the URL that's on the bottom of the page, there is a list of all these, and you will get a copy of these slides emailed out to you at the end of this webinar. So the 1,000 Genomes Project is a large source of our population data. We use the 1,000 Genomes Project for our population data because this is sequencing of a large number of individuals from populations, from healthy individuals in populations all over the world. So the aim of the 1,000 Genomes Project is to provide, what's the word I'm looking for? To provide a kind of catalogue of the base level of normal human genetic variation from different populations. And we provide allele frequencies from all of these. We use these in our linkage disequilibrium. So linkage disequilibrium is the non-random association of alleles at different loci in a specific population. So going back to kind of old school genetics and what we all learned about meiosis, we know that when meiosis occurs, we have crossing over between the sister chromatids. When this occurs, variants which are on the same chromosome can be kind of swapped. And this leads to sort of mixing of different alleles. Where this doesn't occur, what we have is linkage disequilibrium. So if we take this example here where we have two variants, a CT and a CG, we may find that we always get the C and the G and the T and the C occurring together, as in this situation. Obviously, this is too small a sample size to say anything concrete, but we have this individual having just C and G from the two loci. This individual is a heterozygote for both, but you'll see that she still has the same pattern of the C and the G and the T and the C, and this individual is a homozygote for the other pair of variants. But again, it's always the same alleles being co-inherited. You can calculate LD by looking at the frequency that you get these kind of pairings. And it's calculated as the difference between the observed frequency and the expected frequency of all of these different haplotypes. And you get two calculations out of this, D prime and R squared. R squared is, D prime is quite fairly simple because it's the difference divided by the expected difference. R squared is a lot more complicated, and it's to do with correlations, and it's quite a serious calculation to make. The thing to remember is they're both scored out of 1, and if the two loci are co-inherited, i.e. they are in linkage disequilibrium, they will both be 1. And if they are not co-inherited, they're not in linkage disequilibrium, they will be 0. And of course you have a scale all the way between 0 and 1. 0.8 is the usual cutoff that people use for both D prime and R squared to say that things are in LD versus not in LD, but that is something that is flexible. The general way that you visualize, you can visualize linkage is by plotting across a genomic locus, and you use shades of red to highlight the score, either the D prime or the R squared. So the darker shades indicate that things are in linkage, the paler shades indicate that the two variants are not in linkage. And you'll see that it doesn't always perfectly follow a sort of genomic locus of sort of proximity. There are things that can be quite close together, for example here, that are not in LD with each other. So why is LD so very important? Well, if you're looking at disease risk, if you do something like a GWAS, you may find that some of the variants that you will get, some of the loci that will be identified by your study, are actually not the ones that are causing the disease. They're the ones that are in LD with the ones that are causing the disease. So you can get some kind of misleading data from this. So here's an example. In a GWAS of human bitter taste perception, they were looking at common variants in a cohort from Sao Paulo, and they did taste tests where they had stronger concentrations of the chemicals from quinine and caffeine, and wanted to see at what point people could say that's that's got the stuff in versus water. And they did this with genotyping and GWAS. So one of the things they find, so this is a Manhattan plot. This is another way that people show LD, sorry, not how they show LD. This is how they usually show GWAS. So they will show loci compared with the p-value of association. And you can see that the perception to caffeine is shown in black and the perception to quinine is shown in grey. So you've got different variants being highlighted in the two studies. And you've also got a measure here across the locus of the R squared. And you can see the matching of LD of the different variants across this region. So we've got all of the variants in this region that's been shown to have high association with the phenotype through GWAS. You can see there's actually strong LD in all these different populations. So this does kind of mean that it's harder to determine which of these variants are actually causal and which ones might just be tagging along for the ride. They found that there was a cluster of bitter taste receptors on chromosome 12 for caffeine, and they identified a particular variant associated with caffeine and a very different variant, which is very, very close, which was involved in the detection of quinine. So you get very different data for different associations, and it's quite likely that these variants, because of their physical proximity, would be an LD, and you could really mess with your results this way. So we're going to have a look at one of these variants, which was associated with the perceived bitterness of caffeine, and we're going to look at the PEL population from 1,000 Genomes phase 3. So this is a Peruvian population in Lima. And we're going to explore the LD by population of this variant, and particularly looking at this neighbouring variant which was involved in quinine perception. So I'm going to hop out of my presentation. I do have slides of this, so when you download the slides you will get a kind of screenshotted version of this. And I'm just going to copy this variant identifier, and I'm going to go to the Ensembl homepage, and I'm going to put my variant into the search box and hit go, and that takes me to a search result, and I will click on my variant, and I'll just wait for a bit while it decides to load. Right, so now that we're here, it does tell me that this variant maps to 3 locations. I'm not going to go into detail on this. We do have videos on this, but this is just to point out that this is a variant which occurs in a haplotypic region. And when I say haplotype in this case, I don't mean in LD. We actually use the word haplotype to mean 2 different things. I'm just going to click on this one that says chromosome 12 and ignore these 2. They're basically alternative versions of this region of the genome. But I will click on the chromosome 12 and hit go, and now it's going to take me to information about this specific variant. And what I'm going to click on is this linkage disequilibrium button here. So this lists all the different populations that, from 1,000 genomes that I can get data for, explains what the populations are, and I have different kinds of data available to me. So I have a Manhattan plot, I have a table showing variants in high LD, and I also have some plots of LD. So I'm going to go through these one by one and show you what's there. As I said, I'm looking at this PEL population here. So I'm going to start by clicking on View Plot. Just going to open that in a new tab. Right, so we have, this is the location of our variant, and you can see the dots represent all of the other variants in the kind of surrounding neighbouring region of our variant of interest. And we've got a line here indicating, saying 0.8, so this is showing us the R squared of this particular variant, but we can switch this So it tells me it's R squared, but I can change that to D prime if I want to. But these are all the variants which have an R squared above 0.8. So we've got quite a few that are actually on 1. We've got one here that's not quite on 1. And you'll see that actually the neighbouring variants are not in LD. It could be that they have very high or low frequencies. And I'm going to go back to my table that I had before, which I left open in another tab. And I'm going to now look at this variance in high LD page. So I'm going to open that in a new tab. So this actually shows it just at the bottom of the page rather than opening a new page. So it just gives me a list of all the variants and the R squared and D prime values, and tells us which genes they're located in as well. And they're ranked, automatically they're ranked by distance, but you can use these buttons, arrows at the top, to change the ranking. So there's also the LD plot, which is over here. So I'll open that one in a new tab. And this shows us that kind of red, dark, pale plot. So we can see both R squared and further down D prime, indicating which variants are in high LD. So if we follow across the sort of region surrounding this variant, so if we follow this variant down here and then go back up this way, we can see that because there's red here, it's in high LD with this variant over here. I find the Manhattan plots in the tables much more easy to get clear in my mind what I'm looking at. And there's also a table that we can go to as well. So if I click on table from here, it shows me the same data, but as a kind of variance along the top and variance along the side, and indicating it in the same way across a locus with the same style red highlighting. This is a very, very large page. So I find it's not the easiest to find data here. So I'm going to go back to my variant, and what I'm going to explore now, just to show you the population genetics, so you can just get the frequencies of the variant alleles in the different populations. So I'm going to go to population genetics in this page on the left. And this just lists for me in the different 1,000 genomes populations what the frequency is. If I expand, so this AMR is the mixed American. If I expand this out, you can see that there's the Peruvian, the PEL population, and we have the T allele for this particular variant is 90%, and the C is only 10%. Right, so we have the kind of pages in Ensembl where you can view the LD. I had a bunch of slides for this in case it all decided not to work. But we also have a tool, which is the linkage disequilibrium calculator. This is really good if you've got a variant, a pair of variants. It allows you to calculate LD, so from selected populations. So you can get a table with the variant IDs, the location, the consequences, plus the D prime and the R squared. It's currently only available for human data using the 1,000 Genomes Project Phase 3, and it's available in both versions of the human genome assembly. So it can calculate LD across a given region. So if you give it a chromosome, a start and an end coordinate, it will give you LD for all of the variants across that locus. If you give it a list of variants, it will give you pairwise LD for all of the pairs in there. So in this example, these 3 variants out of 5, you can see we're only calculating the LD with those 3. And you can also start at a variant, so you put one variant in, and then you say, right, anything this window size out of it, you can calculate it that way, as in this last example here. So we're going to use the LD calculator to calculate the LD between these 2 variants, which have been associated with perceived bitterness of caffeine and quinine, respectively, again in the PEL population. So if I go back to Ensembl and I go to Tools, up here in the menu at the top, and I click on Linkage Disequilibrium Calculator, this tool here at the bottom of the table, I can choose what kind of LD I want to calculate. So I've got a pair of variants. I'm going to pick LD for a given list of variants. I'm going to give my job a name. I'm going to call it Webinar Demo. And I need to pick my population from the list, which is PEL. And then you've got a box where you can put in your data. So you can paste it in, or you can choose a file, or if you happen to have a file online somewhere, you can put in the URL. I'm going to type in the names of my two variants. So it's RS2708377 and then RS10772420. I'll just check I didn't typo those, because otherwise it won't work. They look correct. So you can separate them in any sensible way that you might normally separate things. I've gone with carriage returns, but commas will work, tabs will work. And it's just it's not put a threshold here for, well, it's put the threshold of 0 for both R squared and D prime. So it will show me everything. It will just show me all of the R squared and D prime between these two variants. If you wanted to do a filter, say if you were going across a locus, you might want to say, I only want to see things that are in linkage. So now I'm going to hit run, and it gives me this job table, and it refreshes every 10 seconds. So it's running the job in the background at the moment for me. It says it's queued, and hopefully it will run relatively quickly. You do have options here, so you can save the job to an account. Oh, it's done. You can edit the job, you can bin the job. You can also share it with somebody. If you copy and paste this URL, you can send it to somebody. But now it says it's done, so I'm going to hit view results. I mentioned earlier about the more than one mapping. So this is what this error message here is showing. It's done the most sensible thing that we expect it to do. So now we have our results. So we have variant 1 and variant 2. We can see that this is a missense variant, and this is an intron variant. It doesn't list the genes. We can get rid of some of these columns, and we can see that the R squared is quite low, but the D prime is actually very high. So depending on the different calculations will sometimes give you different results. So the D prime is telling us that they are in LD, whereas the R squared is telling us that they're not. And I can download this table, which I might want to do particularly if there's quite a large number of them. So that's everything I wanted to tell you about linkage disequilibrium. I'm going to like to tell you a bit about help and documentation. We have absolutely loads and loads of online help and documentation. If you haven't come across EBI Train Online, go there. It is brilliant. It is full of different training courses on using bioinformatics resources from the EBI, but also doing bioinformatic analyses. We have some Ensembl tutorials on there. We also have a YouTube channel, which is where I'm going to post this video with all the fluffs cut out at the end of this webinar. We also have a UQ if you happen to be in China and you want to look at our videos, then you can go to UQ instead. If you are stuck on anything Ensembl related, please email us. We are helpdesk@ensembl.org. You can follow us on social medias. This might have been how some of you found out about today's webinar. We have a Facebook page, a Twitter account, and a blog. And if you are interested in Ensembl training at all, we can teach Ensembl courses at your institute. They are free, other than for the trainer's expenses. We can do browser courses and API courses. If you're at all interested, this site, training.ensembl.org, gives you more information about it, or you can just email us on helpdesk@ensembl.org. We do produce publications. If you use our data, please cite us. This is our team at our most recent retreat, and this is all our acknowledgements, both our funders and all the people who are involved in producing and processing all the data in Ensembl.